For your aquaria setup, you must ensure that all tanks are clean as shown in the video. This is how the setup should look like once it is labeled. For your aquaria setup, you also need to prepare a saltwater solution. First, weigh 30 grams of marine salt using an electronic balance to ensure accuracy. Then, dissolve the salt in 1 liter of heated fresh water. Add the salt gradually to the water to ensure it dissolves completely. The water can be heated using a laboratory hot plate. Once the salt is fully dissolved, transfer the solution into the designated container. This is the designated container that is used for our experiment. This is the brand of marine salt that we use to prepare salt water for this experiment.
Randomization and labeling are vital for scientific accuracy. By using a spin-the-wheel apps, remove bias by assigning treatment to the 9 aquaria randomly. Clear labels like T1A and T2B help distinguish between the 3 replicates for each treatment groups. This organized system prevents mistakes when adding fertilizer and ensures that all growth data is recorded for the correct tank.
In this section, we demonstrate the upscaling technique used in this experiment. Upscaling is the process of transferring algae from small container to a bigger container to support further growth. First, a large aquarium is prepared with 30 ppt salt water. Next, NPK fertilizer is added according to the experimental schedule to provide essential nutrients. After that, algae is measured using a measuring cylinder. The volume of algae used for upscaling is 1 liter. The measured algae is gently transferred from small container to the bigger container to avoid cell damage. After that, the water is gently mixed to ensure the algae and the nutrients are evenly distributed. This upscaling technique helps to achieve suitable algae density and promote healthy growth.
Two fertilizers were used for this microalgae culture, which is the NPK and F2. 3 treatments were applied. Treatment 1 used 100% of NPK, Treatment 2 combined NPK and F2 in equal proportion, and Treatment 3 used 80% of F2 with 20% of NPK. Each aquarium contained 2 liters of algae culture. Fertilizer were added once a week after water sampling. This experiment allowed us to compare the growth response of microalgae under different fertilizer conditions. Daily observations of the algae were recorded using pictures and videos. We monitored the color, presence of bubbles, and overall growth of the cultures. Healthy cultures showed a vibrant green color, which indicates active photosynthesis.
This is the process of collecting sample to be observed using hemocytometer. The algae sample is filled into a test tube until the test tube is almost full. After sampling, each test tube is labeled to avoid confusion during analysis.
After labeling your sample, prepare salt water for dilution. Dilution is only necessary when your sample is too concentrated. To dilute your sample, take 100 microliters of the sample using a micropipette and transfer it into a small beaker. Next, take 100 microliters of salt water and add it into the same beaker containing your sample. After adding the salt water, gently stir the beaker to ensure the sample is properly mixed and diluted. The dilution factor depends on the amount of salt water you add. For example, if you use 500 microliters of salt water, the dilution factor will be 6 as it includes your sample. This means that if you add salt water 5 times, 100 microliter each, the total dilution factor is 6 because you added salt water 5 times and your sample once. After gently stirring the mixture of your sample with salt water, take 100 microliters of the mixture and place it on a hemocytometer. After transferring your sample onto the hemocytometer, carefully place a cover slip to avoid forming bubbles, as bubbles can interfere with your view under the microscope.
After the algae sample was placed on the hemocytometer, The hemocytometer was positioned under the microscope for observation. A 10 times objective lens was used to obtain a clear view of the algae. This is how the algae sample would appear under the microscope. With this sample, we can then proceed with the algae cell calculation.
To get accurate growth data, we use a spectrophotometer to measure the optical density of our algae. First, we must calibrate the machine at a wavelength of 680 nanometer to ensure precise reading. We then carefully fill a clean cuvette with our algae sample and place it inside the machine. We record the absorbent reading and make sure to clean the cuvette between different samples to avoid any contamination. This method helps us confirm our manual cell count and see how the algae density changes over time.
For data recording, all observations were documented in a spreadsheet on a weekly basis. The spreadsheet includes the sampling dates, fertilizer treatments, cell counts, and absorbance values. This organized data log allows easy comparison between treatments and helps to ensure accuracy and consistency throughout the experiment.
In the final part of our project, we analyzed the growth trend of our microalgae over an 11-week period. As shown in our line graph, all treatment began with a similar baseline, but we observed a significant fluctuation in cell density over time. A major highlight of our data is the sharp growth peak in treatment 2 during week 5, although this was followed by a significant crash in week 6 across all groups. This decline likely suggests a period of high nutrient consumption or environmental stress before the next fertilizer application. Based on our experiment, the best treatment is treatment 3 because it shows a more steady and consistent growth trend over the 11 weeks. This combination is also the most cost-effective because it uses a balance of proportional F2 medium and affordable NPK fertilizer.
This graph shows the average absorbent values for 12 samples under 3 different treatments. Overall, the absorbent values vary between treatments, showing that each treatment affects the sample differently. Treatment 1 shows a gradual and more consistent increase in absorbent. Treatment 2 shows higher absorbent at later sample, indicating a delay effect. Treatment 3 shows a sharp peak around sample 5, which is the highest absorbent recorded, suggesting a stronger reaction or higher concentration. In conclusion, treatment 3 showed the strongest absorbent at certain point, while treatment 1 and treatment 2 show more moderate pattern.